Hi children, today we are going to learn about some of the application and the reasoning questions of class 12th biology, principles of inheritance and variation. Okay, and let's see the first question. Human skin color is observed in many shades, many shapes. You know that some are dark, some are fair, some are medium in color and all. Why this difference is there? Because human skin color is controlled by at least 3 pairs of genes. Every gene shows the quantitative effect. So, darkness in the skin is proportional to the presence of number of dominant genes present in the person. Accordingly, these genes express phenotypically. Why can we not take incomplete dominance in Snapdragon as an evidence of blending inheritance? Because reappearance of segregation of both the parents and forms in F2 generation confirms that genes in F1 were discrete. One more question. How is a mutation induced by the mutagen? Explain with example. In biology, mutation is an alteration in the nucleic acid sequence of the genome of an organism like virus or extra chromosomal DNA, because viral genome contains both RNA or DNA as the genetic material. Anything that causes a mutation, that means a change in the DNA sequence caused by mutagens, may be harmful. That means they may cause diseases like cancers. What are mutagens? Examples are radioactive substances, X-rays, UV radiation, and some of the chemicals also. What happens? The mutagen changes the base sequence by insertion, deletion, substitution, and of course addition also. Okay, next question. What is the advantage of presence of sickle cell anemia in heterozygous condition? You know that sickle cell anemia, the genes are in heterozygous condition, HbA and HbS. A group of disorders that cause red blood cells to become misshapen and break down. And with sickle cell disease, an inherited group of disorders, that means red blood cells contort, that means twist or blend into a sickle shape. So the cells die early, leaving a shortage of healthy red blood cells that leads to sickle cell anemia and also can block the blood flow, causing pain. This is sickle cell crisis. And this is caused by the change in the nucleic acid base pairs, that is GAG codes for glutamic acid, GUG codes for valine. GAG and GUG, substitution of glutamic acid by valine is responsible for the sickle cell anemia. So in heterozygous condition, protects the person against malaria. How? RBC having sickle shaped hemoglobin were readily infected with malarial parasite, and with the bursting of RBC, parasite also will die. Advantage. And let's see that why is the woman exceeding 40 years of age have more chances of having child with Down syndrome. What is Down syndrome? It's a genetic disorder caused when abnormal cell division results in extra genetic material from chromosome 21. Not only aged people, children, even the parent is a drug addict or alcoholic, long-term treatments, chronic diseases also will affect the child. So the woman exceeding 40 years of age have more chances of having child with Down syndrome because increased age affects the meiosis of chromosomes and adversely. So the meiosis remains incomplete until fertilization. It remains arrested at prophase itself and chromosome is unpaired. Separation is not done. If the fertilization takes place at this level, and after long gap, the chromosomes will have to remain unpaired for longer time. So the longer the time of unpairing, greater are the chances of its non-disjunctioning, and hence the condition trisomy occurs. One more question. Would emasculation needed for the cucurbit plants. You might have heard about emasculation. This technique we learn in artificial hybridization, emasculation, tagging, bagging, all these are the techniques. So what is exactly emasculation? It's a process of removing anthers from bisexual flowers without affecting the female reproductive part, that is pistil, which is used for artificial hybridization techniques. Okay, cucurbit produces unisexual flowers. That means the male, that is the staminate, and female, that is the pistillate flowers, are born separately on different plants. As emasculation is performed in bisexual flowers, it is not needed for cucurbit plants, but in case of monoecious plants, it is necessary. What are the different steps? Emasculation, tagging, bagging. Then the transfer of pollen grains on the stigma of the other flower, desired flower, and that is the pollination occur. Then the followed by fertilization and flowering and seeding. Fine. The next question. Why is karyotype done at metaphase stage only? What exactly the karyotyping? That means is a general appearance of the complete set of chromosomes in a cell of a species or in an individual organism, mainly including their sizes, numbers, shapes, etc. So, karyotyping is based on size, shape and type of chromosomes. The size is not fixed in prophase, so no chance. In anaphase, the chromatids are not, that means without their chromatids, so there is no chance. So therefore, metaphase is ideal stage for karyotyping. One more question. Can you name any one of the technique, phenomenon, which is exception from the principle of independent assortment? That means independent assortment, all the genes act according to their wish. In dihybrid, you might have seen that 9:3:3:1, but there is a technique, there is a phenomenon which is exception for this one, that is a linkage, because in the linkage, the genes are closely located in the chromosome, so they will transfer as it is to the next generation. And sometimes we see the siblings are having similar characters, not only twins, even some siblings are having same characters, some features are similar. Okay, one more question. At what stage does meiosis occur in haploid organisms such as Neurospora and algae? A haploid organism shows zygotic meiosis. After the meiosis, you can see that diploid condition, the cells become haploid condition and form sperm and egg and all. Okay, but here then fusion of these gametes, the zygote occurs. But here in the haploid condition, haploid organisms, n number of chromosomes only are present. They show zygotic meiosis. Where do you see meiosis? Meiosis occur in germ cells, formation of germ cells, and after zygote undergoes mitotic cell division. But here in the haploid, zygote undergoes meiotic cell division. Zygote is the only diploid stage. Of these, it will undergo meiosis and producing haploid structures which grow to form the body of haploid organism. That's the answer. Okay, next question. In one experiment of mitosis, scientists wanted to arrest the cells at metaphase stage. This happens once they want to produce more number of cells in this particular phase. This may happen in vitro fertilizations. This may happen even artificial hybridization. So what do they use? They use a chemical colchicine. That means COLCHICINE. And next question. What is a tetrad analysis? Tetrad analysis, ability of manipulating and individually study the four products of a single meiotic event. That means it has been critical to understand the mechanism of heredity. So, in the examination of all the products of an individual in order to find out the distance between the two genes in the linkage and recombination frequency between the two genes, segregation of the traits, all these will come under this tetrad analysis. What is chromosome map? By finding recombination frequencies for many gene pairs, we can make linkage maps that show the order and relative distance of the genes on the chromosomes. This will understand the linked genes and how they transfer from one parent to the offspring. Thank you so much and have a nice day.